Welcome to this explainer. Today, we're diving into the microscopic world inside our cells to uncover the unsung heroes keeping us alive. We're looking at something truly fundamental to our biology, a system so elegant and so vital that without it, life as we know it simply wouldn't exist.
Think about it for a second. Right now, every second of the day, your cells are churning out thousands of proteins, these incredibly long, complex chains of amino acids. So, what keeps them from just tangling up into toxic microscopic clumps? Because if these chains fold incorrectly, the results are literally catastrophic. The stakes couldn't be higher. So, who exactly is stepping in to manage all this microscopic chaos?
Well, here's our roadmap to figure that out. First, we'll meet these cellular guardians and see exactly how they protect us. Then we'll look at the frontline heat shock families, dive into the cell's built-in quality control system, and explore the diseases that happen when these guardians fail. Finally, we'll wrap up with how biotechnology is harnessing all of this for the future. Let's get right into it.
Okay, Section 1. Meet the cellular guardians. We're introducing the protein protectors.
So what are chaperone proteins? Basically, they're specialized proteins that assist other proteins in achieving or maintaining their correct structure. But here is the absolute crucial detail. They do this without becoming part of the final product. They're like the ultimate biological guides, right? They don't provide the final structure themselves, they just step in to ensure the protein folds properly, prevent any dangerous interactions, and then boom, they step away once the job is done.
And we need this guidance because, honestly, protein folding is an unbelievably complex process. Newly synthesized proteins pop out of ribosomes as just these loose, floppy, unfolded chains. To actually do their jobs, they have to fold perfectly into highly specific three-dimensional structures. If they don't, they're useless. Or worse, they're actively dangerous. Chaperones are those essential workers making sure every single protein reaches that perfect 3D state so our cells can survive.
Moving on to Section 2, how chaperones protect us. Let's look at their 5 critical functions.
Think of this as their core job description on the cellular factory floor. First up, assisting protein folding. They literally bind to newly formed proteins and guide them into shape. Second, preventing protein aggregation. They spot abnormal proteins and stop them from clumping together into toxic messes. Third is refolding. If a protein gets damaged by heat or stress, chaperones act as a repair crew to fix it. Fourth, they handle transport, safely escorting proteins across cellular compartments, like from the cytoplasm over to the mitochondria. And fifth, well, if a protein is just too damaged to save, they provide degradation support, marching that ruined protein straight to the cellular trash.
All right, Section 3, the heat shock families. These are our frontline responders.
Now, these frontline responders actually get their name because they multiply like crazy whenever the cell experiences stress. Think a sudden fever, an infection, or oxidative damage. Let's look at the most studied family here, HSP70. Think of HSP70 like a cellular mechanic. In step 1, it identifies and binds to the unfolded, exposed regions of a newly synthesized or damaged protein. In step 2, it grabs some ATP, the cell's primary energy currency, to power up. Step 3, our mechanic actively wrestles the protein into folding correctly. And finally, step 4, the beautifully repaired, correctly folded protein is released back into the wild to do its job. It's a brilliant 4-step mechanism, and it is happening millions of times inside you right this second.
But HSP70 isn't flying solo. We've also got HSP60, also known as chaperones, which actually form these tiny, barrel-like structures to give proteins a private, isolated VIP folding room, totally away from the crowded cellular chaos. Then there's the HSP90 family, specializing in folding proteins used for cell signaling and hormone receptors. Oh, and we definitely can't forget these small heat shock proteins, or SHSPs. These are like emergency stabilizers for damaged proteins to stop them from clumping. A really cool example of this? Alpha-crystalline, which literally keeps the proteins in the lens of your eye clear and functional so you can see.
That brings us to Section 4, the quality control system. Let's talk about maintaining cellular balance.
So, one of the most vital alarm bells in this whole system is the unfolded protein response, or UPR. Now, the endoplasmic reticulum, or ER, is a massive protein-producing factory in your cells. When it gets backed up and overwhelmed with an accumulation of misfolded proteins, it essentially hits the panic button. The cell activates the UPR to freeze general protein synthesis and just massively ramp up chaperone production to clear out the backlog and save the cell.
But, what if the protein is just totally beyond repair? Well, that's when the ubiquitin proteasome system kicks in. This is the execution phase. Step 1. That irredeemably damaged protein gets tagged with a molecule called ubiquitin. Basically, it gets slapped with a biological trash sticker. Step 2. The proteasome, which is essentially the main cellular shredder, recognizes that tag. And step 3, the protein is fed right into the proteasome and gracefully dismantled back into basic amino acids so the cell can recycle them. Super efficient.
Sometimes, though, the garbage is just way too big for the shredder. If massive toxic clumps form, the cell has to use autophagy. This is a heavy-duty cleanup system that acts like a cellular garbage truck, specifically designed to swallow up larger protein aggregates, damaged organelles, and huge protein complexes. It keeps the whole cellular environment clean and functional.
Okay, Section 5. When chaperones fail. Diseases. Let's look at the high stakes of misfolding.
Now, what's really interesting here is what happens when this incredible system actually breaks down. We're faced with this fascinating medical paradox. On the left side, you have neurodegenerative diseases, things like Alzheimer's, Parkinson's, and Huntington's. These are characterized by a failure to clear misfolded proteins, causing toxic aggregates to build up in the brain. So medically, our goal here is to desperately increase chaperone activity to clear those clumps out. But look at the right side, cancer. Cancer cells produce massive amounts of abnormal proteins and live in a state of constant stress. To survive and grow so rapidly, they actually hijack and boost their own chaperone production, specifically HSP70 and HSP90. So for cancer, the therapeutic goal is the exact opposite. We want to inhibit chaperone activity to starve and stress the tumors to death. It's a real high-wire balancing act.
Finally, Section 6. The future of biotechnology. Engineering our own guardians.
Researchers are already putting chaperones to work across the biotech landscape. In recombinant protein production, where we basically use bacteria to manufacture proteins for us, chaperones are solving the huge problem of incorrect folding, which drastically improves both stability and production efficiency. In drug discovery, chaperones are prime targets for those cancer and neurodegeneration therapies we just talked about. And in protein engineering, they are absolutely essential for producing complex industrial enzymes and engineering totally novel proteins.
But the bleeding edge of this research? It's honestly mind-blowing. We are looking at developing specific chaperone-based therapies to artificially enhance our natural protein repair systems. We're seeing targeted protein degradation tools, things like PROTACs and molecular glues, which allow us to intentionally hijack the cell's tagging system to aggressively hunt down and remove specific harmful proteins. And perhaps the wildest part? We're now using artificial intelligence to design entirely new, superior chaperones from scratch. We are creating folding assistants and stabilizers that nature never even dreamed of.
So, to bring this all together for you, chaperone proteins are the overarching guides here. You've got HSP70 doing the direct repairs, HSP60 providing the private folding chambers, and HSP90 stabilizing signaling proteins. When things fail, the proteasome shreds the small stuff, autophagy swallows the big stuff, and the unfolded protein response acts as the master alarm bell for the ER. Together, they form the ultimate cellular quality control system.
And it really all comes down to this. Chaperone proteins are the essential guardians of cellular health. They are quite literally our frontline defense against cellular aging, catastrophic breakdowns, and devastating diseases.
Which leaves us with a pretty massive final thought to ponder. If these microscopic mechanics are already doing so much to keep us alive right now, what incurable diseases could we conquer if we successfully engineered AI-designed chaperones to perfectly protect our cells? The blueprint is already inside us. Now, we just have to learn how to master it. Thanks so much for joining me on this explainer, and keep questioning the microscopic world within you.